Okay, micro chapter 3. To be honest, there won't be a lot of detail in this chapter for the quizzes, and I'll explain why in a little bit. Let's There we go. Sorry about that. Wait a minute. Sorry. Hold on 1 second. All right, sorry about that. I couldn't find a PowerPoint. Observing. Oh, okay. So basically, okay, unaided eye can only see so much. Okay, light microscopy, okay, allows us to see things that are smaller, and then even smaller than that, you need electron microscopy. Electron microscopy uses electrons instead of light. The wavelength of light is too big, so it can jump over things. Okay, and so we use electron microscopy to see. Okay. All right. So a microorganism is usually measured in micrometers, okay, otherwise known as microns or nanometers, okay. So this letter is actually the Greek letter mu. So 1 mu m is 10 to the minus 6 meters. So literally 1 millionth of a meter. Okay, a micrometer is 1 millionth of a meter. Okay, or 1,000th of a centimeter. Okay, 10 to the minus third millimeters. So 1,000th of a millimeter. 1 nanometer is 1 billionth of a meter. Okay, 10 to the minus 9th meters, or 10 to the minus 1 millionth of a millimeter, or 1,000th of a micron. Okay. Okay, so make sure you know how to do problems like this. Okay, the answer, okay, is 10,000. If you got that, then you're on good track. Because there will be something like this on the test. Okay. Don't worry about diagramming the length of light through a microscope. Okay. And the microscope will be more of the lab exam more than anything else. But I do want you to understand what the total magnification is. Okay. So in compound microscopes, there's always at least two, well, compound microscopes by definition have two lenses. Okay. One at the top near the eye is called the ocular lens, and one near the objective, or one near the actual thing called the objective lens. Okay. The total magnification of that is the two numbers crossed together. So if both lenses are 10x, then the total magnification is 10 times 10 or 100. Okay. Okay. Resolution is the other scientific term you have to understand. Okay. So I want you guys to do something. Okay. Imagine yourself on a railway. The rails are obviously equal distance apart, right? So you're standing in the middle of a railway and you're staring off into the rail. What you'll see eventually is the rail will get narrower and narrower and narrower. And at some point, those two rails will merge into a single rail. The distance at which that occurs is the resolution that you have. Okay, the shorter wavelength of light, okay, actually provide greater resolution because it jumps over less things. So blue light, purple light, UV actually has greater resolution. Okay. All right. Refractive index is the measurement of light bending ability of a medium. Light may reflect after passing through a specimen to the extent it does not pass through objective lens. And that is why we use, for the 100x scope, we use immersion oil to allow the light to travel directly through instead of refract off. Okay, the cool thing is the reason why the immersion oil is the immersion oil and the glass slide have the same refractive index almost, and they should, in theory, have the same refractive index. Okay, and so you can see the light pass straight through that. Okay, another way of putting it this way, this is actually a cool thing to do if you have enough immersion oil. If you take a glass slide and you drop it into immersion oil, like the slide will disappear because it'll literally have the same refractive index, so you'll see right through it. Okay. All right. We'll skip the bright field resolutions and stuff. Okay. The light microscopy stuff. We'll skip as well. Okay. I'll skip most of this stuff. That's for lab. Fluorescence microscopy. This is fluorescence microscopy. So we attach using molecular biology techniques. We can attach something called GFP, green fluorescent protein, to cells in the organism, and then by using light that reflects off of GFP, we can make these things glow and make them really easy to see. Okay. Confocal microscopy is really cool, but we'll skip that. Two-photon microscopy is even more cooler. When I first started grad school, they actually shoved me in a two-photon microscopy lab, and they were like, "Oh, this microscope is worth $250,000, so don't fuck it up," which terrified me to death. And it's $250,000. It's a freaking house. And if I screw it up, that would be very bad. But I didn't. Okay. I will skip most of this. Okay. Electron microscopy, that I do want you guys to have a pretty good understanding of. We're shooting electrons instead of light. So again, these things, you'll never see in a regular college level microscopy class. So they cost probably $100,000 a scope. Okay, they shoot electrons through and electrons bounce off. There are two types of electron microscopy. One is called scanning electron microscopy. Okay, and you can see this, and you can see a lot of structures here that you can't see under a light microscope. But what makes it extra cool is the transmission electron microscopy. Okay, transmission microscopy. You see how this it looks? It's the same organism, but it looks much more 3D. Okay, so scanning electron microscopy is one more expensive. Two, you have to work. The organism has to be dead beforehand, but there's much more 3D structure. Okay. No more about tunnel and all this other stuff. Okay. Here's where you do have to know some stuff. Okay. Okay. So you have to differentiate an acidic dye from a basic dye. Okay. Explain the purpose of simple staining. Explain a gram stain. Okay. All right. And like all these. Okay. So staining. Okay. Microorganisms are usually not particularly colorful, right? They're not, I mean, they don't really want to attract attention to themselves, so they're not usually colorful. Staining, you color microorganisms with a dye that emphasizes certain structures, okay? Um, a smear is you spreading a thin film of substance over a slide. The microorganisms are fixed and attached to the slide, which kills the microorganisms. Okay. Okay. Live and unstained organisms have little contrast with the surrounding medium. The live specimens are used to study behavior, and you kind of see them when you look at them under a microscope. You kind of see them, but not you don't see them in any great amount of detail. But you can see how they move and stuff. When you stain microorganisms, you usually end up killing them. Okay. Uh, okay. I'm not sure if you guys heard that, but I'll explain it. Okay. Um, so stains, okay, consist of two parts. Okay, a positive and negative ion, one of which is colored. Okay, in a basic dye, the chromophore is a cation. Okay, so the color thing is a positively charged thing, so it attaches to things that are negatively charged. Okay, the color attaches to negatively charged things. In acidic dye, the chromophore is an anion, so it attaches to positively charged things. Okay. Okay. Staining in the background instead of cell is something called negative staining. So your cell comes across white or clear in a background of black. Okay. Okay. A simple stain is a use of a simple dye. You add color to the cell. Okay. So um, okay. So the negative stain stains the outside of the cell, right? So okay. Fixing is necessary because you have to so the way you do a stain, okay, is you add the stain to the slide, and so the whole slide is stained that color. What you do next is you have to wash the stain off, except the ones that are attracted to the cell. And to do that, you have to wash it. And if you wash it, you have to make sure you fix the microorganism to the slide, or else it'll wash off when you wash. Okay. All right. Um, Differential stains are used to differentiate between different bacteria. I know that sounds, simple, but it's actually not. Okay. The first stain is called a Gram stain. Okay. So gram-positive bacteria have a thick peptidoglycan cell wall. Gram-negative bacteria have a thin peptidoglycan cell wall. And a layer on top of that is called lipopolysaccharides. Okay, so um, the first thing you have to do is fix the organisms to the slide. Okay, that you if we do micro lab, you actually burn, put it across a flame and like seal it into the slide. The next thing is you add crystal violet stain. Okay, the crystal violet stain will enter the cells and it will stain every single cell. So if you stop right there, you can't tell the difference between gram positive and gram negative. Okay, the next thing you do is you add iodine. Iodine is called a mordant. The purpose of a mordant, it attaches to the stain, and now the stain is bigger, and so it can't move in and out of the cell quite as easily. In fact, the iodine stain renders it incapable of moving out of the gram-positive cell wall. Okay, and then you wash with alcohol. The alcohol will wash off the free-floating iodine. It will wash out the crystal violet stain if it's not attached to iodine, but it will not wash off a combination of iodine and crystal violet in a gram-positive cell. Okay, that way, at this stage, what you'll see is just the gram-positive cells are purple. And then you put a counter stain. The counter stain, okay, is the saffron. The saffron will stain everything pink, all the cells pink. The gram-positive cells, you already have purple. So adding a little bit of pink to purple does nothing. It just makes it more purple, but they're still purple. The gram-negative cells now pick up the pink. Okay. Okay. So now the gram-negative cells, okay, will be pink, and so now I can see a very clear difference. The gram-positive is purple. The gram-negative is pink. Okay. So you can see a real one right there. This is a really well done one. Usually they're closer in color. Okay. An acid-fast stain binds only to bacteria that have a waxy material in their cell walls. Okay, which is not decolorized by acid-fast alcohol. Okay, so you add a primary stain. Again, that stain stains everything. Carbofusion stains everything. Acid alcohol in combination washes out the red stain, except when that cell is surrounded by wax, like a thick layer of fat. The acid alcohol can't get through the fat, so it doesn't wash that out. So now only the acid-fast bacteria are still red. So this is now colorless. Okay, and then I add a light color stain. The light color counter stain is methylene blue. Methylene blue, again, will stain everything blue, but the light blue that methylene blue is on top of the red will still show up red. Methylene blue on top of nothing will show up blue. So the non-acid fast bacteria will show up blue. The acid fast bacteria will show up red. Okay. Remember, the counter stain stains all the cells, but if you already have a color, you're just adding more color, and that allows you to tell one color apart from a second color. Okay. This is actually important because this mycobacterium, that is the bacterium in one of the biggest diseases, TB, tuberculosis. So using acid-fast staining, we can readily identify TB. Okay. Okay. The special stains will skip. Okay. Okay. All right. So we will. Well, the special stains we'll go over in lab, but not lecture. Okay. So I will stop this presentation right here, and I will see you in chapter 4. Chapter 4 is a great deal more complicated than chapter 3, unfortunately. Okay. So if you have time today, start on chapter 4. Okay.